Hi, the other preprint that's out today. This is another update on Flower Jungle Friday. Is from the Barrick Lab, and what they've done is in this preprint, they have made a modified version of SARS coronavirus 2, which is good. And what they've done is they've adapted it. So there are two parts of this one, two amino acids in spike and that aren't making very good contact, or that work well enough with a human but do not work particularly well with a mouse. And so they're changing this thing so that we can actually get this virus to grow in mice. Because right now, SARS coronavirus 2 is not good at growing in mice. You just don't get an infection with mice. And so once we have a mouse model that we can work with, that's much better. The way people normally do this is by putting the human receptor protein or proteins into a mouse. And the problem there is that the proteins don't necessarily get expressed in the right places. So the mice get a disease, but it's not the same disease. And so there are some problems with, you can show that you can cure them, but they're not really facing the same thing as a human would be. This, though, is a modified version of the virus, and the changed version seems to grow very well in mice. And they've got it growing in young mice and old mice. And so they can potentially do experiments that look at, the dependence of age, how age affects how the virus gets you. So that's kind of neat. The two amino acids that they changed are number 498 and 499 in the spike protein. I think they were a glutamine. They were a Q that changed to a phenylalanine, if I remember right. Did I get that right? No, a threonine. Yeah, bugger. Yeah. But also a proline at 499 that changed to a tyrosine. And these are just amino acid names. There's 20 different common amino acids, and we're just swapping out one and putting in another. The swaps were based on the structures that we already have of the way that the spike protein interacts with the receptor, the ACE2 receptor. And they were also based on molecular modeling studies where you say, okay, so we'll substitute in the mouse version of the receptor, and the spike now doesn't bind very well, but if we change this, this, and this, then boom, we can get a really good binding face. That's the idea. That's what they did. It seems to have worked, at least as far as I can see in the preprint. I'm curious to see what, if anything, changes when this thing actually comes out, because I feel like this is going to be coming out fairly soon in an actual journal in finished form. But useful. I think a lot of labs may want to use a model like this. The virus that they're making it's not more dangerous for humans. It would be more dangerous for mice. So this is something that would have to be used inside the laboratory, under the normal precautions that we take in a laboratory, which are really high compared to the precautions that the average SARS-infected person takes when they go to the store, which, are usually really low. I think this can be useful in helping scientists figure out exactly what it is that we should be looking at and how we can fight this virus. So, it's a pretty good thing. So there we go. That's a Dr. Ben update. Thanks very much.